Alright, let's open the right page. The mantis shrimp. The mantis shrimp is not a shrimp. It is not a mantis. It is a stomatopod, a lineage of marine crustaceans that diverged from the main crustacean family tree approximately 400 million years ago and has been evolving independently ever since. What that 400 million year independent development produced is a striking appendage, the dactyl club, a calcified hammer-like structure folded against the front of the body and used for both predation and defense. In peacock mantis shrimps, which reach approximately 18 centimeters in length, this club can accelerate from rest to approximately 23 meters per second in 27 milliseconds, an acceleration comparable to that of a .22 caliber bullet leaving a gun barrel, achieved within a body length of a few centimeters. The impact force generated is approximately 1,500 newtons from an animal that weighs grams. A 2004 study published in Nature by researchers at the University of California, Berkeley, and the Field Museum measured these values directly using high-speed videography and force sensors. The physical impact alone would be enough to shatter the shells of most prey species. But the strike generates a second, independent destructive mechanism, cavitation. As the club moves through water at that velocity, the pressure behind the club face drops so rapidly that the water locally vaporizes into cavitation bubbles. Those bubbles collapse milliseconds later, with temperatures briefly approaching 4,700 degrees Celsius at the collapse point, and shockwaves that deliver an additional impact to the target independent of the club contact. The dactyl club absorbs enormous repeated impacts without fracturing. The pistol shrimp. The pistol shrimp, any of approximately 600 species in the family Alphidae, takes a fundamentally different approach to predation than physical impact. It has evolved a specialized asymmetric claw with a geometry that, when snapped shut, displaces water so rapidly that it creates a cavitation bubble in front of the claw tip. The collapse of that bubble produces a shock wave measurable at 218 decibels at close range, louder than a jet engine, louder than a rifle shot, and louder than almost any biological sound source documented in nature. The thermal spike at the collapse point of the cavitation bubble reaches approximately 400 degrees Celsius for a duration of less than a millisecond hotter than the surface of the sun briefly. In a volume smaller than a cubic millimeter, the shockwave stuns or kills small prey fish and crustaceans at close range. The snapping mechanism works through a specific claw geometry. A plunger structure on the moving dactyl fits precisely into a socket on the fixed propolis. When the muscle contracts and the dactyl snaps shut, the plunger socket fit creates an explosive pressure gradient that drives the water jet and generates the cavitation. The acoustic output of pistol shrimp colonies, and they live in colonies, collectively snapping thousands of times per second in reef environments, is substantial enough to have caused operational problems for military acoustics during World War II. Submarine commanders discovered that their passive sonar systems were being masked by the constant background crackle of pistol shrimp colonies across the shallow coastal waters of the Pacific Theater, making it difficult to distinguish submarine acoustic signatures from the biological noise floor. Naval acousticians eventually cataloged the source. The tardigrade. Tardigrades are microscopic animals, typically between 0.1 and 1.5 millimeters in length at maturity, that inhabit almost every moist environment on Earth. The water films on soil particles, freshwater sediment deep sea floors, Antarctic ice sheets, hydrothermal vent fields, mountain snow fields at altitude, and the surfaces of mosses and lichens growing on nearly every continent. Their survival capabilities operate at the boundary of what the physical and biochemical parameters of life have historically been understood to permit. When environmental conditions become incompatible with normal metabolism, extreme temperature, desiccation, ionizing radiation, vacuum, toxic chemistry, tardigrades can enter a suspended metabolic state called cryptobiosis. In the specific form called anhydrobiosis triggered by desiccation, the animal retracts its limbs, expels nearly all body water, and synthesizes a glassy disaccharide called trachelose at concentrations sufficient to replace the water structurally, vitrifying the cytoplasm, immobilizing biological molecules in a solid glass matrix without allowing the destructive ice crystal formation or protein denaturation that would otherwise result from extreme drying. In this tune state, metabolic activity drops to below 0.01% of active levels, and the animals can remain viable for decades. Tardigrades in ton form have been revived after exposure to temperatures of -272 degrees Celsius, 1 degree above absolute zero, for extended periods in laboratory conditions. They were exposed for 10 days to the open vacuum of space combined with full-spectrum solar ultraviolet radiation on the outside of the International Space Station during the Photon M3 and EXPOSC missions in 2007 and 2008. and the majority survive to revive and reproduce. The immortal jellyfish. Turritopsis darnii is a small marine hydrozoan, typically 4 to 5 millimeters in bell diameter, found primarily in the Mediterranean Sea and in tropical ocean waters globally following likely human-assisted dispersal through ballast water. It is the only known multicellular animal capable of complete biological age reversal, not extending lifespan, not slowing aging, but reversing the developmental trajectory and beginning again from a juvenile stage. The mechanism is transdifferentiation, the conversion of mature, differentiated adult cells directly into different cell types or into an undifferentiated state, followed by redevelopment, under conditions of starvation. Physical damage, disease, or aging. The mature medusa form of Turritopsis darnii sinks to the seafloor, reabsorbs its bell and tentacles, and reverts to a cyst state from which it develops back into a sessile polyp, the juvenile form it occupied before its initial transformation into a free-swimming adult. The cells of the bell and mesoglia dedifferentiate, muscle cells lose their contractile architecture and become pluripotent, nerve cells revert. The polyp then develops forward again, differentiating into a new medusa indistinguishable from the original. The process can repeat indefinitely in laboratory culture. No upper limit on cycle number has been documented. In practice, wild Turritopsis individuals are subject to predation, disease, physical damage, and ocean chemistry effects that prevent indefinite persistence in nature, but the cellular mechanism itself appears to have no intrinsic cycle limit. The genes responsible for Turritopsis' transdifferentiation capacity include several that have homologs in mammalian cancer biology, specifically oncogenes associated with cellular dedifferentiation and proliferation. The lyrebird. The superb lyrebird of southeastern Australia, Menura novaehollandia, holds two distinctions that together place it in a biological category no other animal occupies. It has the most anatomically complex vocal apparatus of any bird species, consisting of a specialized syrinx with musculature capable of producing independent, simultaneous control over multiple pairs of sound-generating membranes across a frequency range from approximately 1 to 12 kHz. And it uses that anatomy to produce exact acoustic replicas of sounds it encounters in its environment, not simplified imitations or approximations, but spectroscopically verified reproductions that are in many cases indistinguishable from the original source by acoustic analysis. A lyrebird will learn the calls of neighboring bird species through sustained exposure, eventually reproducing them with a frequency precision and temporal accuracy that researchers using sonogram analysis describe as acoustically identical to the source recordings. Lyrebirds have been documented producing the exact calls of kookaburras, eastern whipbirds, gray striped thrushes, and complex multi-species flock choruses rendered simultaneously. They have reproduced the mechanical sounds of chainsaws, camera shutters, specifically the distinct mechanical sequence of a camera mirror flip, aperture actuation, and shutter close, car engines, and construction equipment. In documented cases from forests in New South Wales, where logging occurred during the 1930s and 1940s, lyrebirds in populations that had no current contact with machinery continued to reproduce chainsaw and axe sounds that had been incorporated into the local population's song culture and passed culturally between generations of birds for decades after the original sound ceased. The axolotl Ambystoma mexicanum, is a salamander native to the ancient lake systems of the Valley of Mexico, specifically Lake Xochimilco and the remnants of the broader Aztec lake network that covered much of the Mexico City basin before colonial-era drainage projects. It is best known for a developmental characteristic called neoteny. Unlike most amphibians, which undergo a metamorphic transition from an aquatic juvenile stage with external gills and a lateral line to a terrestrial adult form with lungs, the axolotl retains its larval morphology throughout its entire life, breeding in a form that is physiologically equivalent to a permanently juvenile salamander. But neoteny is not the scientifically significant characteristic. The regenerative capacity is. Axolotls can completely regenerate functional limbs following amputation, not through scarring and approximation, but through full morphological restoration, including bone, muscle, cartilage, vasculature, skin, and peripheral nerve tissue with correct positional patterning and full neurological integration into the spinal cord. The process takes between 2 and 4 weeks depending on the size of the lost limb. The regenerated limb is functionally and structurally correct. The regenerative capacity extends beyond limbs. Axolotls have been documented regenerating substantial portions of ventricular cardiac muscle following surgical excision, segments of spinal cord tissue following transection, and areas of retinal tissue following injury. The mechanism proceeds through the formation of a blastema, a mass of locally dedifferentiated cells accumulating at the wound site. reverting to a stem cell-like state under the influence of molecular signals from the wound environment, which then redevelops under positional guidance signals from the surrounding intact tissue. That's it for today. Subscribe for more, smash the like button, and drop a comment with your thoughts. I read every single one. Peace.